Welcome to Project Here, speaker series presentation featuring Scott Long. My name is David Vera. I'm an associate professor and chair of the Department of Economics at California State University, Fresno, and member of Project Here's executive committee. On behalf of the co-directors, Richard Ball and Norm Medeiros, and the members of Project Here's executive committee, I'd like to welcome you to today's session. Today's presentation is the fifth of 9 weekly webcasts that will feature leaders in transparency and reproducibility movement. We're truly excited to offer this series and would like to thank the Alfred P. Sloan Foundation for the generous sponsorship and continued support of Project Here. I would also like to thank Alex Sabath at Haverford College for coordinating the technical aspects of this series. In case you experience audio or video difficulties during the broadcast, please know that we'll be recording today's sessions and posting it to the Project Here website for later viewing. And now to the reason you're here, it's an honor to introduce today's speaker, Scott Long. Professor Scott Long is a distinguished professor emeritus and Chancellor's professor emeritus of sociology and statistics at Indiana University. In his methodological work, he has developed new methods for the interpretation of statistical results, unified literatures from diverse fields, and developed methods to facilitate reproducible research. He's the author of texts on confirmatory factor analysis and structural equation modeling, regression models for categorical outcomes, and methods for reproducible results. He taught statistical methods at Indiana University and for the ICPSR summer program. His substantive research examined gender differences in scientific careers, health among mid-life women, stigma and mental illness, and human sexuality. This work was supported by the National Institute of Health and the National Science Foundation. Dr. Long was awarded the American Sociological Association's Paul F. Lazarsfeld Memorial Award for distinguished contributions in the field of sociological methodology, and the Limer Rosenthal Prize for Open Social Science. He's an elected member of the Sociological Research Association and an elected fellow of the American Statistical Association. Professor Long will present for about 45 minutes, after which we'll have a few minutes available for questions. Throughout the presentation, please type your questions in the Q&A panel on Zoom. I'll assemble the questions and post them to Scott as time permits at the end of his talk. So without further ado, let me turn the broadcast over to Professor Scott Long. Thank you.
I'm setting up the share here. All right. Well, thank you for that introduction. And I'd also like to thank Richard Ball and Norm Medeiros for all the work they do at Project Here to make this possible. My talk today is about a computing workflow. Basically, a workflow for computing is a set of strategies that allow you to efficiently and accurately obtain results that can be reproduced. Now, to put my talk within the broader context of this series of lectures, I want to refer back to the wonderful talk that Michelle Newton did 2 weeks ago on the 4 steps for efficiently checking robustness of results. In the first step, she talked about checking the internal consistency of results. The computing workflow contributes to this because many of these consistencies occur from errors that have been made in computation. The second step involves using the same data to reproduce results using the original methods, something I think that was referred to as exact reproduction. My talk, if you will, is a precursor to this. It is how you can reproduce your own results with your own methods. After that's accomplished, it's possible to go to these more advanced methods of reproduction using alternative methodologies or using alternative data sets. So the question that I'm addressing, if you will, is what I call internal reproducibility. Can you, the researcher who's published a paper, reproduce your own results using your own data and your own analysis scripts. If you talk to someone who doesn't do quantitative research, it may take a while before they understand why this is so difficult. All you're being asked to do is redo what's already been completed. In my experience, many researchers have difficulty doing this. Files are missing, or the wrong versions of files are available. Their scripts no longer run, or if they run, they're providing different results. To give you some evidence of the prevalence of this problem, the American Journal of Political Science has the most stringent requirements for reproducibility of any journal I've seen. They ask authors or potential authors to submit data sets and scripts before review. Before the review process begins, they have students work on these scripts to make sure that they can reproduce the results. Early on, of 200 submissions, less than 5% were able to confirm the results. That is, the authors submitted scripts and data sets that didn't work. Eventually, almost all of these authors were able to provide scripts that confirmed the results, but the point is, many authors do not have results that they can replicate themselves. Now, to assess whether this is an issue for yourself, a really useful exercise is to take a paper that you wrote, ideally a year ago or more, but it can even be last week. See what it takes to rerun your analysis and to exactly reproduce your findings. Ideally, you can do this with making no changes to your scripts or your data sets. When you're done with this, give your files to a colleague and ask them to do the same. Here are a couple of examples of things that happen. All of these are examples that I've been involved with in one way or the other. A retraction that occurred when a data set was lost, or the data set that a person had was the wrong version. A researcher spent the 2 worst weeks of his life trying to reproduce the results because he feared he was going to have to retract an article. Eventually he succeeded, but the initial scripts weren't working. A student was defending the dissertation and couldn't show where the results came from. It took him 9 months to write the dissertation and do the initial analyses, and 18 months to work backwards to show where those results came from. I've encountered collaborators where they're both analyzing the same data set. It has the same name, but they differ. Consequently, the results are inconsistent. I've seen in published papers or in the review process, the same variable in the same table was created differently in different scripts. Or when I was invited to work on a collaboration to enhance the methods for a paper that was drafted, I was given a single 743-line script file. It generated hundreds of lines of output, thousands. None of the results from the paper were reproduced. Essentially, we started over. So what the computer workflow is designed to do is to minimize these sorts of issues. It sets up a series of strategies that reinforce one another, that allow you to compute in a way that's more efficient, more accurate, and more reproducible. Now, I refer to this as a computing workflow because there's many workflows that are possible. And here are the criteria I use when I'm choosing. The primary criterion is reproducibility. You need to make sure that however you compute, you can reproduce the results. Next, you want accuracy. It doesn't do you a lot of good if you can reproduce the wrong answers. So you want to prevent both minor and retraction-worthy errors with your workflow. Efficiency. You can't reproduce a paper that's not published. You've got to get the work done as efficiently as possible. You'd like procedures that are scalable. That is, you can use the same procedures on small projects and large projects. You want the procedures and strategies to be adaptable, so they're not dependent on a very specific computing environment. You'd like them to work if you're using the supercomputer or your laptop, Unix, Linux, Mac, whatever it is. You want them to be adaptable. And ideally, you want them to be accessible, particularly if you're involved in collaborations. It does very little good to have extremely demanding strategies that require advanced technology if your collaborators are not comfortable with it. So the workflow involves 6 basic components. First is the use of script files. Second is the notion of posting. You never change files that were used to produce your results. A dual workflow in which you separate the data management from the data analysis. Something I call run order naming. Your scripts are named so that if they're run in alphabetical order, they reproduce the results. The use of effective names and labels so that your output is clear and understandable and so that errors are prevented in specifying models. And the construction of a reproduction package that includes all the scripts and data sets that are necessary to reproduce your results. So I'm going to go through each of these in some detail. Now, before I begin though, I want to emphasize that no workflow is perfect. No workflow is going to be optimal for every situation that you're going to encounter. As you look at the things I propose, you're going to find that some of my strategies are, to put it mildly, irritating. They seem like an annoying thing to have to do. But many of them are designed to prevent errors, problems that can be devastating, even though they're rare. Trying to avoid those things that are unlikely to happen, but if they do, they lead to things like retractions. You also want strategies that prevent problems. In my experience, fixing problems ends up taking far more time than adapting a workflow that prevents them. And I would encourage you to make exceptions reluctantly. It's easy to say things like, this time I'm not going to post the files because I'm really sure I'll never use them again. You're never really sure. Okay. Scripts. Scripts are simply text files, ASCII files that run your software. Sometimes they're called do files in status, syntax files, code files, program files. They're simply the commands that run your software. Now, why use scripts? Scripts are by far the easiest way to obtain results that are reproducible. Friends simply don't let friends point and click. It's a great way to sell software. It's easy to learn, but it's really hard to use. Scripts also provide exact documentation of exactly what you did. The easiest way to tell someone what the statistical procedure is that you used is to tell them the software you used and send them your scripts. Finally, scripts simplify correcting errors and making revisions in the future. Now, for this to work, you need scripts that are robust. That is, they need to run on a different computer at a later time with no changes. They also need to be legible, so they're easy to understand. So let's think about making robust scripts. Completely robust scripts that are going to run in the future are simply impossible. The software I used to write my dissertation doesn't exist anymore, and there's no way to get those scripts to run. But in the nearer term, you want scripts that have several characteristics. First of all, you want the script to control everything that affects the results. For example, it should load the data set, not assume that the data set is in memory. It should set any parameters that affect the results. It should not rely on things that other scripts left in memory. Okay, so some software, R and State in particular, one script often depends on results that are left in memory. Okay, that makes the script fragile. Here's the most common example I've seen. The first script uses the variable income. It creates income squared, but it doesn't save that variable to a data set. The second script is fragile because it assumes that inc squared is in memory. One of two things is going to happen. If inc squared is not in memory, the script won't run. That's okay, because then you'll fix the problem. More dangerously is when the wrong inc squared is in memory and the results are wrong. So it's just a simple example of not relying on what's in memory. Another common example involves random numbers. Random numbers are used for a wide variety of aspects of statistical analysis. These numbers are generated by setting a seed. It's just a number. That number is used to generate a string of random numbers. The results you obtain can vary greatly by the seed that is set. To be robust, then, you need a script that sets the seed. To give you just a simple example, I generated 5 different random samples from a data set and rerun stepwise regression on all of them. I took the most extreme cases. In one case, we find a strong suppressing effect of being a female on the incidence of diabetes, and in the other script, there's no effect. Without knowing the seed, I'd have no way to reproduce those results. Okay. Second issue, your results should not depend on your computing environment. The most common example of this is when authors of scripts hard code information about their directory structure. The directory structure is the drive letters or the directories that you're using. In my environment, the drive letters on the local area network change periodically when systems are changed. If I use a different computer, my laptop has different drive letters than my desktop. And if you're sharing files with others, you can't assume that they're going to use the same directory structure. So to illustrate this, if you use a command like this that's labeled fragile, it only runs if the person who is using it has a directory structure, D drive, user directory, username, JS long, groups, work, and there's my data set. What you want is a robust command that simply uses the data set and assumes that it will be found in the default location. As an example of this problem, the director of statistical consulting at Indiana sent me a copy of an email that was received, where a paper was submitted to a journal. The editors sent it back on review, and it said, your script sets the directory. Please resubmit your code without an absolute path. What they were saying is, your program won't run on our computing system. Now, no matter what you do, your results will depend on the software that's being used. Accordingly, to have reproducible results, you need to either provide the software that produces the same results, or you need to present the exact details on how the computations were made. Problem one that commonly occurs is that user-written commands have to be installed. This is a very, very common problem in my experience with R, where there's many user-written packages, or Stata, where there's also a very vibrant user community. So for example, I wrote a command called mchange with Jeremy Fries. If you install the package, the command runs fine. If I send the script to someone and they haven't installed the package, they're going to get an error that that command is not recognized. So you have to make sure that users know exactly what scripts are installed. A second problem, new versions of software can give you different results. This happens commonly when a new command is being introduced. Future releases of the software might introduce new algorithms for more efficient or more accurate results. The syntax of commands might change. This is very commonly a problem with user-written scripts. And commands might change what they leave in memory, so a script will be fragile if it depends on this. Other software disappears. At one point, BMDP was a very common practice, and it's completely gone at this point. You also will find that different file formats disappear. Osiris was extremely common at one point. It's virtually gone at this instant. So what you need to do is your script should indicate exactly the software that's being used. I'm using Stata 16, it's 64-bit version, 8 cores, and I'm using the release on the 11th of November, 2019. Then indicate all of the user-written packages that your scripts depend on. That way, both you in the future and other users have a better chance of making your scripts run. Now, what I like to do with the completion of a paper is to make a tarball. That is, I bring together all of the software that's required to run the analysis of my paper, and I make a backup copy of it that I put in deep storage. If that's not feasible, at least back up the user-written packages so you have them in the future. Another thing that's incredibly valuable is to save your data set in multiple formats. That way, if a particular format like Gauss format files is no longer available, which is now the case, you'll also have the data in other formats. Now, how do you know if your script's robust? It's pretty easy. Go to another computer and try to run it. If that works, ask a colleague at a different computer to run your scripts. And we'll talk about this later when we get to what's referred to as a reproduction package. Legible scripts are pretty simple. You want scripts that are easy to understand. This helps you avoid some errors and makes it much simpler in the future when you come back and need to fix a script. I suspect that many of you have encountered the problem that you've gone back to something you did and you can't figure out exactly what was going on initially. Here's some examples quickly. Without alignment, it's really hard to see errors and follow it. Simply by aligning commands, it's much easier. When you're running a logit, I've had people send me scripts that look like this. It's very hard to check the variables, but simple formatting makes it much easier to see what's going on and prevent errors. Another example is to make sure that you can see the entire command. I'm surprised with how many people write incredibly long commands. A user of one of my commands sent me a script, and their command is written here for you. It's about 200 characters long, and what I could read was truncated at 80 columns. The command gave peculiar results that I could not figure out, and eventually I reformatted it and immediately realized the problem was the user had thrown out the intercept. Think of this as running a regression where you force the intercept to be 0. You're going to get peculiar results. Another thing that makes your scripts much more effective and legible is to add comments. Indicate, for example, the degree variables indicate enrollment, not completion. This would have saved me a month when I was working on a project with other researchers. Indicate that the sample is restricted to the paid labor force. If you're doing preliminary analysis, add a comment that says this is the partial data of a particular date. If you're using procedures that are unusual or require special justification, add a reference that justifies what you're doing. If you find results that are surprising, add a note that indicates you verified these, and ideally add some comments as why the result may be occurring. Limit abbreviations. Here's one of the favorite ones I've ever seen. This is a completely valid command that stands for list age and labor force participation in these cases. They're quick to write, but they're very hard to understand. Use names and labels that are effective. Names like RO215 are very difficult to remember what they are, and it's easy to make mistakes. When you create variable labels, remember they get truncated. This is an actual example from a project I was on. How important is it to go to, turn to? All the useful information was truncated in the output. Or even worse, here was a scale that went from definitely agree to definitely disagree. And with truncation, all you know is you have definitely and definitely, and it took 3 PhDs to argue about which category was agree. Okay. Make sure your output is legible, not truncated. On the right, not wrapped, and don't print markup language as straight ASCII. Okay. Then, when you're done with your scripts, refine them. Verify the accuracy, improve the legibility, refine the comments, and confirm that the script runs. The next principle is what I call the posting principle. This is a form of versioning. It deals with the problem if you don't have the files, you won't be able to reproduce the results. Posting is defined by two very simple rules. Only share results after the files are posted. Secondly, never change a posted file. Here's the way this works. Let's suppose that you do all of your work in a directory called work. This is where my scripts are, my data set. This would be the working directory that I use in Stata. When I think that the work is done, I'll put it in a folder called review. This is called staging. It sits there and I let it age. It gives me some time to return to it at a later point. I recall the work to my work directory, verify that things are all right, and then I move it when I'm satisfied to the posted folder. So before I share it and when I think it's done, I post the results. Once the files are in posted, they never move back to work and review because those files are essential for reproducibility, so you can't change them. What you can do is you can make copies of them with different names. So let's suppose that I posted a file called data do, and it created a data set. And later I discovered that one of the variables was incorrect. So I would take the posted data script, posted data1.do, I would copy it to the work folder and change the name by adding v2. Then I would change the second version of the script file to create a second version of the variable that's saved in a second version of a data set. I have to change the names because I can't post them with the same names because there's already files there that have been posted. What posting does is it makes it much less likely that you're going to use a critical file, and it prevents you having a file with the right name, but where the content was changed. If you keep changing a data set without changing the name, your results become incredibly fragile. It also has other benefits. You know where your critical files are located, so backing up is simpler. The progression through stages reduces errors because it's more thoughtful. It documents a file's status. Anything in posted you know is complete and there's nothing else to be done for it. And backing up is much simpler because the only things you really have to back up are those that are posted. Now, I've preached this for many, many years, and I've had graduate students send me messages like, well, the data sets are exactly the same, except that I changed the married variable. To me, it's not exactly the same. You changed the posted file, or I didn't think you'd use the data set, so I changed it last night. Again, violating this notion of posted, both cases led to inconsistent results. The next strategy that I recommend is to differentiate between data management and data analysis. For me, data management is less exciting, more exacting, and more fundamental. If your variables are incorrect, you're not going to fix them with the analysis. Analysis often sparks creativity, and that creativity, for me at least, encourages poor data management. I'm anxious to do the analysis. I'm less worried about labeling variables and verifying them. So what I suggest is that you have a data workflow that creates variables. It verifies the variables, labels them, and documents them. It selects the observations, and the data sets are saved and documented. The analysis workflow uses data sets, but does not create them. It uses variables, but does not create them. So for example, on the left is my data workflow. I take my source data. I have two script files that lead me to the first analysis data set, Russia1. Russia1 is used by two analysis scripts. One does descriptive, one does some ordinal logits. I decide to add some variables. I create Russia2. I use that for a couple of other analysis scripts. I decide I want to add some IRT scales. I add another script that creates another data set, and that's being used by scripts over on the analysis workflow. The critical thing you notice is no data sets are being created on the blue side. So what's the advantage? For me, the advantage is that errors are much less likely from careless data management. You're unlikely to create a variable and forget to put it in a data set. You're more likely to verify variables, to label them correctly, and it also makes it simpler to go back and correct errors. If you're involved in collaborations, I find this is a much better way to avoid different collaborators creating data sets that are in conflict, that you have the distinction and it's easier to share the files and to revise them. Now, there's some disadvantages. You have to create a data set when you're certain you will only use the variable once and it doesn't need to be in a data set. In my experience, you never really know this. Reviewers have minds of their own, changes get made. It's best to just assume that it's going to happen. Secondly, it's annoying to interrupt data analysis to create a data set. It kind of gets in the way. And the third problem is you're much less likely to have your paper advertised on retractionwatch.com because it's going to prevent a lot of errors. Now, how can you make this dual workflow simpler? This is what I find works the best for me. Before I begin data analysis, I write a cloning script. This creates my working data set. It is simply a copy of my latest version of the analysis data set. It's exactly the same, but I give it a new name. This working data set is used by my analysis scripts. When I need a new variable, I go back to the cloning script, create a new variable, run it again. It recreates the working data set. Since that data set hasn't been posted, there's no problem. My analysis script continues to work on the working data set. I can continue to do this, and as I need new variables, I continually add them in the cloning script. When analysis is complete, I can look at the cloning script, go through, verify it, add metadata, do all the things I want to create a new data set, rerun the analysis scripts, and it takes very little extra time over creating data sets in your analysis scripts. Okay. The next strategy. The next strategy is not really required, but it's convenient. Run order naming is the notion that you name your scripts so that they should be run in alphabetical order. So if you look over here at the example we had, you run explore first, then extract, then add bin, then add IRT. How do I remember that that's the order? I could rely on my memory, which isn't going to work too well. I could write documentation, but then I have to refer to that and write down trivial sorts of things. I could try it different ways to see what works. I could look at each script and figure out the order. I could use a master script, that is a script that runs other scripts, or I could use run order naming. So here's what run order naming does. Before I proceed, I meant to point out the data scripts have to be run in a specific order. So for example, you can't create Russia 2 unless you have Russia 1. You don't get Russia 1 unless you run extract 2. So you have to work in exactly this order. The analysis scripts can be run in any order as long as the data sets exist. So I could replicate my results working from the last file to the first. They'd all run as long as my data sets exist. So let's look at our example of using the run order naming. Without run order naming, here's how my scripts are going to appear in my browser or in my file manager. They're just a bunch of alphabetical files. In a large project, there may be dozens of scripts. With run order naming, I might simply say, I'll stick data 1 in front of the name of the first data script and then adds explore data 2, data 3, data 4. My analysis scripts are just numbered in the same way, so I immediately know the order in which the scripts were written and the order in which they need to be run. For the analysis scripts, even though they could be run in different orders, I personally find it very useful to know which results were created most recently. What was my last analysis? What were my first analyses? Okay. This doesn't affect reproducibility, but it's simply a matter of making your workflow more efficient and to provide ways to organize your research more effectively. One of the ways in which the efficiency shows up is when you find an error. So for understanding this, you want to think about two types of scripts. You have upstream scripts. These are scripts that can't be affected by the error. So going back, we know that if there's a mistake here, it can't affect Russia 1 because it doesn't have anything to do with creating Russia 1. So extract do and explore do are upstream. The later scripts could be affected by what happens in add bin. So let's suppose that I create data1.do through data9.do, and I create 9 different data sets. I find an error in my 6th script that created the 6th data set. The upstream scripts, data 1 through data 5, that created the first 5 data sets are fine. I know I don't have to check them. The downstream scripts could be affected, so all I do is I take the posted scripts, I copy them to my working directory, rename them to version 2, make the corrections, rerun them, and post the results. What's nice is it's very simple to be able to tell which scripts are going to be affected, and it makes it easier to determine which of your analysis scripts might be affected by errors in your data set. Okay. So to this point, I've been talking about things you do when you're writing your scripts to make them more effective. Okay. At this point, I'm imagining that you have a draft of your paper. You've got the scripts, you've got the data sets, you've extracted the information, and you put them in a paper. At this point, you should create a reproduction package. What this package is a set of files that let you reproduce your results. You include files with documentation that describes your software. It might add information on variable construction, sample selection, data sources. You include all of the scripts that were used to produce all data-driven results in your paper. Anything that came out of your data should be included in these scripts, and the data sets that at the minimum allow your analysis scripts to run. Now, for data sets, there's much more complexity as to what your reproduction package needs to include. At the minimum, it needs to include the variables, and observations that were used in the analysis scripts. But this is limited in that other people can't check the robustness of your results or see how you created your variables. At the other extreme, you can include all of your source data sets and or information about how to obtain them, since sometimes you can't distribute source data sets, and include data scripts that transform the source data sets into the analysis data sets. This is the requirement of the American Journal of Political Science. You submit your source data sets, everything that was used to create your analysis data set, and all of your analysis. Now, there's some constraints in providing all of your data sets. Most importantly, some data sets have restricted use policies, so you're not allowed to distribute them. Okay, this has to be honored. A more practical problem is that researchers are often reluctant to share all of their data when they haven't finished analyzing it. So this is something in which the community is still debating what's the best way to proceed. An issue that you can consider, though, is when should you create your reproduction package? What I find is the most efficient in the long run is to, when I am writing the paper, think about creating the reproduction paper. Keep track of all the data-driven conclusions. Keep track of every variable and observation used. Then, before you share a draft of your paper, create your reproduction package. It gives you a chance to go back, look at your scripts, recreate your data set, and one more chance to correct errors. I had an opportunity recently to do this, and in the process, we discovered a problem while creating the reproduction package. So it's just a great idea, even if you're not ready to distribute it. Then, before you submit a paper for review, update the package. Some journals only request the paper or these files when the paper is accepted. It's much easier to have it ready before that. Other journals will only ask for such a package when your results are being challenged and they want to provide your files to another author. So for me, I find it much, much better to create this package as you work and to make it available to other researchers. It's certainly part of open science. It also improves the reproducibility of your work. Okay, so let's review what I've been recommending. Okay, the computing workflow is something that supports reproducibility. It also, I believe, increases your efficiency in the long run. By long run, I mean, it may take you longer to write a script, but if problems are found, it's going to be much quicker to fix them, and it's also going to prevent errors that can be catastrophic. Many of the retractions that I have seen involve simple errors in the data analysis that could easily have been prevented. When you find the errors, the workflow makes it much simpler to make those fixes. The notions of posting, that whenever you share results, you can never change the files that were used to create them, is in my mind absolutely essential for reproducibility. Posting, in effect, provides what is available through something like GitHub. It's a much simpler way to do versioning that doesn't require the substantial overhead of GitHub. The other strategies involve efficiency and accuracy. It's possible to reproduce the wrong results. That's better than not reproducing them, but you'd really like to have the results to be the correct answers to the questions that you're asking. And you want to work efficiently. Unless the paper is published, there's nothing to reproduce, so a computing workflow will make your work go faster. There's also some other advantages. I find the computing workflow is particularly useful when you're revising a paper. In my experience, reviewers always want changing, and if your script files are well organized and legible, it's much quicker to make those changes. When you're working in collaborations, the workflow makes it simpler to coordinate the work among the different authors. It also makes backups much easier since you know exactly which files are critical. And non-trivially, I also find that the workflow reduces stress. And that, for most of us, is a very valuable contribution. All right. Thank you. Questions?
All right, well, that was an excellent presentation by Professor Long. So I got a question that we can, actually a couple of questions that sort of will be straight, I guess you will be able to address following your work. So the first one is from Christopher Conway. For the absolute directory paper that the editor returned unreviewed, what would be an example of how you would write in a relative directory path?
Right. It varies across packages, but most packages, statistical packages, if you try to load a data set, will look somewhere by default. You set the default directory. In Stata, it's called the working directory. So what you would do is always load data from the default directory. Once you do that, the problem disappears completely. In some packages, you can also use what are called relative directories. So it doesn't know the drive letter, but relative to the default directory, it's in a subdirectory called data, for example. So this is something that you can check within your different software as to how you do it. Okay?
Excellent, excellent, excellent. So I have another question from Kate Doyle, and actually this is one that I was thinking about. So Kate asked about what are your thoughts on using dynamic reproducible documentation, you know, like R Markdown in R?
Yeah, things like R Markdown and other packages have gotten a lot of attention, you know, reproducible results and dynamic documents. For the most part, I don't find that it makes a huge contribution for the kind of work I'm doing. That is, if my program won't run in the future, it doesn't matter that now what doesn't run is something that created dynamic documents. Secondly, a real critical problem is none of the packages that I have seen let you extract the results in exactly the form that you want them in. So let's say you take something like Stata that has dynamic documents, you can grab all of the regression results, but they're not in the form that you want them in the paper. One of the consequences is that the tables are oftentimes not as effective. You can actually tell when this is how they were created. A third issue is I think dynamic documents encourages people to make rapid changes rather than more thoughtful changes. I don't make that many revisions of papers. It won't save me the time to do it for the most part. Creating tables takes a lot of time, but it doesn't take me a lot of time to put them into my paper because I'm not going to make revisions that often. So those are different aspects of it. So you can tell it's not something I found terribly useful. With one exception, I worked with someone who has to put out a weekly report where the core of it is similar. In that case, it was great to set up a very effective and sophisticated generation of the tables for a dynamic document. Then they just went in and wrote the text around it, and it saved them an immense amount of time. But that's kind of an exception.
Excellent, excellent. Okay, I have a question from Richard Ball. He's interested in your views on how well the principles you have presented are integrated in the training of social scientists. So, I mean, specifically, how about sociology?
Yeah.
Do undergrads get taught this stuff? About graduate students, is this widely considered to be bread and butter stuff that must be included in quantitative method courses, research, or something on the fringe?
First of all, it's changing. I found that when I started classes by showing slides of retractions in the journals they're hoping to publish in, people paid a lot more attention to reproducibility and correct results. Retractionwatch.com is a great place to encourage people to take these things more seriously. But by and large, we don't teach it. And you know, I think Haverford has done this amazing job introducing it as an undergraduate curriculum, which is really where it needs to start. When I taught a course on reproducibility for master's students, it included students both in the statistics department and sociology. The statisticians hated it. They really didn't see what's the big deal. I just want to run the analyses that are sophisticated. And so what it showed is the students going through statistics weren't really given the motivation for reproducibility. The social scientists were more interested, and the more senior the student, the more interested. That is, reproducibility and these principles become much more interesting when you have a disaster, when your paper's been challenged, when your dissertation is refused because you can't show where the results come from. So it's something that is starting to change. Sociology in particular, I think, has been the worst of the disciplines in making the push. It's only this year that the Methods Conference is focusing on reproducible results. Other disciplines, I think, have been a little bit more advanced. Sociology is always contrarian about this. But no, I find that it's a difficult process. What I've started doing in graduate stat classes is I require everybody to submit all of their script files for all of their assignments, and I have the teaching assistant rerun them. If they don't run, you lose 25% of the grade, which gives you a failing grade. The first time students are outraged, and I simply say it sure beats a retraction. The second time, their scripts run. And I think that's part of the training that becomes required to make this happen. So Richard, I definitely don't think that it's an easy sell yet at this point.
So I actually have a question regarding big data machine learning. So anyone that is doing data analysis nowadays, of course, is either getting into big data or working with big data, and the idea of using machine learning methods, and that could be at very different levels. As they become more widely used for research, particularly machine learning, what do you think the role would be for transparency and reproducibility? What role would they play? Sometimes, I mean, in the context where you have these somewhat black box type of analysis when it's machine learning based, what's your thought about that?
You know, working with students in information science, I've seen that there is a bit of black box, and that there's all kinds of challenges. For example, I had a student who was working on data that was streaming in real time. There was no way to keep that data. So, but what could be done was to keep a precise record of exactly what was done. A critical aspect of that, particularly in fields like machine learning, where the algorithms are being updated so regularly, is things like GitHub or packages like Sumatra that allow you to reestablish exactly the code that was used so you know how it was produced. So I think that's where it tends to be particularly important. And even if you don't keep the data, if you're able to give someone the scripts, so you know the data set's too big to preserve, fine, I still could look at your scripts to know what you did and determine if there's problems. The same thing I think applies if you're using confidential data sources. For example, parts of Ad Health cannot be put in the public domain. I find that that's just fine, and those data sets should not be distributed, but the authors should be asked to present their scripts to create the variables, their scripts for the analysis, so others can evaluate how the work was done. Okay. So I think in all these contexts, the notions of reproducibility are helpful. It ought to at least be the instruction so someone could go out and do the same thing again.
Okay, I think we have time for one last question, and I think Richard, he's just submitted another question. Can you point to instructive examples of any published papers for which authors have made excellent reproduction documentation publicly available, either voluntarily, so on a personal website, or because it was required by a journal?
Yeah, I think the place that I would go as the model for this is the American Journal of Political Science. They have a model that really won't scale because they got funding so that if I'm an author submitting a paper with my source data sets and scripts, they ship the data and scripts to North Carolina at the Research Institute there. I've lost the name for a moment, where they have hired graduate assistants who rerun all the scripts before the review process. These scripts are made available to the reviewers along with the paper. And they have pretty detailed instructions on what they're asking people to do. I mean, that certainly is the model. My experience has been that in a paper recently, they only wanted them. They wouldn't take them until the paper was in press. Prior to that, they didn't want to see them. And I think that's really a mistake. I think all journals should at least require authors to deposit them prior to the review process, and indeed make them available to the reviewers, even if they don't look at them themselves. But there aren't a lot of models that I know of this.
Excellent. Well, I think this is a good place to close the webinar today. And I would like to thank you all for attending today's session, and a very special thank you to you, Scott, for providing such an informative presentation. I hope you will tune in again next week, same time, same channel, for Dorothy Bishop's webcast entitled Why Scientific Reasoning is Hard, the Role of Cognitive Constraints in Biasing Our Reasoning. Again, thank you all for attending.
Let me thank everyone who attended. This is my first webinar or use of Zoom for a lecture, and it was a learning experience for me for sure. So thank you all. I hope you found this useful.
Thank you.
Goodbye.
Bye.